We are pleased to have Karen Gulner as our presenter. Karen is a registered pharmacist, has a PhD in pharmacy from Otago University in New Zealand, a postdoc at Boehringer Ingelheim Respiratory Delivery Group, lecturer at Massey University, manager global clinical trial suppliers at Boehringer Ingelheim Vetmedica, product support engineer, and is now a product manager at Agilent. All right, it gives me great pleasure to turn today's event over to Karen. Karen, please take it away.
Yeah, hello, thank you. So first thing I have to do is share my slides. Hang on. There we are, and put them in presentation mode so you can see them properly. There we are. So the Dissolution Toolkit, so everything you need from innovations to data integrity. So we're gonna kind of whirl through this array of topics today. So we're gonna talking about really is the I have to get this box out of the way. So the dissolution new developments, performance testing of novel dosage forms, then dissolution qualification is a hot topic, satisfying the AIQ requirements for your dissolution apparatus, and then data integrity, creating a paperless compliant dissolution testing environment. So that's going to be our three main topics. Let's get started. Excellent. So the slides are not moving. Now it's working. Okay. So I'll give you a short introduction on the dissolution topic. So the dissolution test really has evolved to become a definitive tool to characterize the performance characteristics of many types of pharmaceutical dosage forms and not tablets alone. And the dosage forms, they have become more varied over the last 50 years, as we all know, and the dissolution apparatus has required really continuous improvement and modification to provide suitable conditions for performance testing. So we cannot test all our dosage form with our beaker and pedal apparatuses anymore. So dissolution is an important tool for characterizing the biopharmaceutical properties of a pharmaceutical product at different stages throughout its life cycle. So during product development, you will do API characterization, formulation evaluation, and stability testing. And already for the formulation evaluation and stability testing, dissolution tests will be required. Then for the bioavailability research, you will do in vitro and in vivo correlation, and also further bioequivalence studies, of course. And in quality control, it's really more of a pass-fail product release. But once you even have released your product, you will continue to need dissolution testing for the scale-up and post-approval changes. So if you change your raw materials, if you change your formulation, the process, the manufacturing process or your manufacturing site, you will do further dissolution testing and development. So dissolution, as I said, assesses the performance of drug products in your body, basically, or we try to do that, mimic it here in the vessel. And to be very effective, the test should be predictive, comparative, discriminatory, and reproducible. Right? So these are the goals we are aiming for. And as I mentioned, dissolution is not just about orally ingested products like tablets or capsules. Tests must be also done on suspension, powders, coated beads, ointments, transdermal patches, microneedle patches, wound care products, powders for inhalation, chewing gums. So everything that has an API in its formulation and is not a true solution needs dissolution testing, basically. That's kind of a rough guideline there. And now you have to imagine that you need a kind of dissolution tester to accommodate all these different formulations, right? So what are the challenges apart from the new formulations? So there are analytical challenges with small volumes, right? If you only have a couple of drops that are, drops is a bad example, but only if you have really low API concentrations that are in your formulation, you will run into analytical challenges with the quantitation limit. You all know what it is. It's the lowest amount of analyte in a sample that can be determined with acceptable precision and accuracy under the stated experimental conditions. So the reason for that is that you also see now LC-MS methods for the analytical finish of your dissolution samples. So we had small volumes, then low dose formulations, the oral dosage units containing concentrations of your API at microgram or nanogram levels, even ocular or other mucous membrane direct applications really usually are low dose formulations, or inhalation products often also have a low dose. Or even if you have a very prolonged release, I think that's coming on the next slide, yeah, so if you have long-term extended release formulations, you have continuous release of low concentrations, days, weeks, or months even. That often happens with implants and depots, with transdermal microneedles or drug-eluting stents. We have quite a lot of experience with these, the drug-eluting stents and the low concentrations, or even bone cement, just another example there. So further challenges, you have rapid release formulations. How do you analyze your amount of API there? For immediate release, sublinguals, gel films, very rapid release, but still you can set up a dissolution testing method for that. Everything is possible, basically. The next challenge is the poor solubility of your API, so you have to look for sink conditions. That can be very challenging. I mean, how big can you go with your volumes, right? If you need multiple volumes or continuous flow even, right? All things you could consider. So what's the variety of dissolution and drug release apparatus? Of course, I'm sure you're all familiar with the apparatus 1 and 2 that you see at the upper bit here. Usually you work in 1 liter vessels, but even 2 liter and 4 liter vessels are official, right? So if you're running into sink conditions, or if you have a veterinary product, then these 4 liter or the 2 liter vessels are very popular. We even have a bespoke system made for us with 5 liter vessels. Everything is possible there. That's not compendial anymore, but it is possible. The operational minimum in your 1 liter vessel is about 400 milliliter for our system. What you would normally test in your standard apparatus 1 and 2 really is your tablets, your capsules, suspensions, suppositories, chewable tablets, and even powders. Transdermal systems, you would go for apparatus 5 and 6. Basically, it's the same instrument, but you have, instead of pedals, you have, for the apparatus 6, you have a rotating cylinder. For the apparatus 5, you actually do have a pedal, but you have a disc lying in the bottom that contains your transdermal patch. You can automate your apparatus 1 and 2 with UV or even LC. So what can we do for novel dosage forms in our apparatus 1 and 2? We have a modified stationary basket. So you utilize the best of both worlds. You utilize your pedal, and you have a stationary basket that goes through your evap cover where you have your formulation in there. Then there's a basket that's also the USP for fallopian extended release tablets. And for these tablets, it's very important that they have a certain direction when you have them in the vessel to get a good release profile. So now when you are sort of running into major challenges with your apparatus 1 and 2, you would go to the next instrument, which is the apparatus 3. You have about a volume like the vessel size of 300 milliliters, but your working volume is about 175 to 275 milliliters in volume. And you can see already the major difference in the instruments is you're not steering a pedal or a basket, but you are dipping up and down, right? Very different motion. You can also see that you're not just staying in one vessel, so to speak. Of course, you can do that. You can run your whole dissolution test in just one row. But normally you would sort of go and utilize an apparatus 3 if you, for instance, want to change your media. So you want to start with a very low pH here in the first row, so probably to mimic the stomach. And then you could theoretically move through your whole intestinal system by moving from one row to the other, right? So what do you usually use? Sort of what type of formulations? You have extended release tablets, capsules, beads, chewable tablets with glass beads. You can even that to mimic a kind of chewing motion there, right? And modifications have been made to the instrument, so you can go up to even 1 liter vessel in that system. Now, your dosage form would be contained in one of these glass cylinders, and normally you would have a mesh at the top and the bottom of that cylinder, so your formulation, your dosage form doesn't fall out of that cylinder. So what have we tested? This is just a picture here of some nasal sponges that we tested with the apparatus 3. Just for more novel dosage form, here is an oral patch holder that can be attached to the apparatus 3. What else is there? There is the apparatus 4. We at Agilent don't have that system, but just to mention it here, it was initially designed for poorly soluble extended release compounds, and typical flow rates from 4 milliliters per minute up to 16 milliliters per minute. You can have a closed system with a small media reservoir, and you can go down to volumes of about like 30 milliliters, but you can also have an open system where you are pumping fresh medium in all the time, and your formulation would sit in one of these flow cells here, and everything is then driven by a pump. So quite sort of similar to what you use the apparatus 3 for: extended release tablets, beads, suppositories, and implants. You have some special cells for powders and suppositories. Sort of maybe a little bit on the historical background. Why don't we at Agilent have such a system? Well, Agilent is an American company, and we acquired the Dissolution product range when we bought Varian. So Varian also is an American company, and the apparatus 3 was developed in the US. It was also called the ELSA apparatus for these kind of applications with extended release tablets, beads, this kind of things. And the apparatus 3 was developed in Switzerland by the competitor Sotax together with Novartis. So it's a bit of like a regional historical kind of thing where these apparatuses reside, really, right? And both, I think, have their justification. So the apparatus 7, as you see it here from Agilent, looks very similar to the apparatus 3. The difference is it only dips for 2 centimeters. The apparatus 3 dips for 10 centimeters. Also, you have smaller volumes, right, as you can see on the picture. And the great thing about it, and what I really like, is you have sort of the freedom of choice of your holders that you can use in the apparatus 7, right? So there's a whole array of holders that you can see down there on the picture. And some of them are also described in the USP. But basically, you can also make your own holders if your holder doesn't exist, and you need something else, and you can justify the use. So what it's used for: also extended release tablets, capsules, beads, implants, transdermals, osmotic pumps, stents, and other novel dosage forms, and with a smaller volume, really. So if you have to go even lower in your volume, you would go away from your traditional apparatus 7 to a 400DS, which you can see on the picture here. So you can go down to volumes as low as 3 milliliters. This instrument was designed by Agilent, or then Varian, for drug-coated stents. That's what it was initially designed for. Now, these dissolution tests for drug-coated stents, they run for a very, very long time. So the instrument had to have excellent evaporation control. And you can use some organic solvents as media even, also sort of due to those great evaporation control. And what you have in the bottom is also an automated sampling and media sampling system, and you can replace your media throughout your test. So you can connect up to 5 different media bottles to that valve up here and exchange media during the tests, right? And you don't have a water bath. You have dissolution cells sitting on this holder here, and they are surrounded by a heater jacket. So you have very tight temperature control, and you can actually heat up your system up to 55 degrees at very great precision. And we have customers doing this to accelerate their dissolution testing. Again, here is another slide. Yes. So the application, really, your drug-eluting stents, as I mentioned, that's what it was originally designed for. You have these holders here that you can see on the picture on the right. Microparticulates, you can use it for medicated contact lenses, pacemaker, electrodes, and also your micro needle patches, right? Again, you are free to choose your holder. We can make holders for you. You can even make them yourself. The whole thing is driven by a magnet, so that is sort of the only requirement. You have a magnet sitting there in the top that then moves your holder up and down. So what are there as modifications for your standard dissolution apparatuses? You have the vertical diffusion cell. There are sort of 3 different versions described based on the classic Kranz cell. I don't want to go into too much detail because we don't sell them. They're not used a lot, but you can basically have them here for your reference, right? What we do have and what customers, what we get sort of more questions for is the immersion cell method that is used with a special vessel that you can see here on the picture and a mini mini pedal sitting on top of it. So this is really sort of for your creams and ointments, right? And you have then a definitive surface over which your formulation will release the drug, right? And you work with a membrane in between. It all has a definitive size there. So what else is there? It's a flow-through method. I mentioned that already. Then what we also see as a trend really is the mini pedal and the mini basket. So we are using our standard dissolution tester and have adapters for smaller vessels. So for 100 and 200 milliliter, we also have a 250 milliliter vessel, which is from the Chinese pharmacopoeia. Again, it's used for tablets, capsules, powders, especially if you have low API concentrations, right? So inhalation products. What's interesting, there are currently no requirements for dissolution testing of particulates from MDI or the DPI aerosol products. But various devices have been used for research purposes. So there's a powder cell in the apparatus 4, which uses the whole formulation. What you do is you collect fractions from the Anderson cascade impactor, and filters from each fractions are then placed between membranes in a special cell for the apparatus 4. And using the NGI apparatus, also special collection cups are covered with a membrane, and the dissolution is then measured in apparatus 2. So you can use that device and put it in your standard dissolution tester in the bottom, a little bit like the pedal over disc assembly there, right? So let's come to quite a different topic, and that is satisfying your analytical instrument qualification requirements for the dissolution apparatus, right? So the USP requirements for pharmacopoeial dissolution tests were first introduced in 1970 for 6 monographs. That's when it started. And sort of the basic concept of your dissolution apparatus was really sort of established by empirical means rather than sort of sound scientific and engineering consideration. And it was pretty obvious that the different apparatuses produced very different results, right? So often these apparatuses were custom made. The control of your mechanical parameters that was inadequate, and vibration was also a key factor that couldn't actually be quantified at the time even. And so the use of a calibrator was suggested, right? So what you have to do, I'm sure you all know that Chapter 711, determine the acceptable performance of the dissolution test assembly periodically. The suitability of the individual apparatus is demonstrated by the apparatus suitability test. So this apparatus suitability test that has sort of been subject to extensive review over the several years, and it has changed quite significantly. And the USP revised the concept of the test from a test on each individual position in a dissolution tester to the ISO approach of instrument proficiency testing. We're going to see in a minute why. So that your result or your apparatus suitability is based on the whole instrument rather than the individual position. And that was introduced as the performance verification testing. So for the performance verification testing, it's a statistical analysis of the results of tests on all positions of an apparatus are now required, not just a single position, all positions, and both the mean of the results and the percentage coefficient of variation must pass the acceptance criteria. And in line of that, the prednisone tablets, they were renamed as reference standard tablets to remove the implication of calibration, right? So you cannot calibrate a dissolution tester with tablets. Is just not possible. So the problem with the original requirement to evaluate individual positions was really like, what do you do if you have like results that all kind of center around this sort of 30 to 35% area, but then you have this great outlier here, right? I mean, is this still acceptable? Obviously not, right? So what else can we say to the PVT? So the PVT is really a holistic test and provides experimental proof that the dissolution test assembly is suitable for dissolution testing. PVT was designed as a single-stage test using the ISO approach for instrument proficiency testing, which means like 2 consecutive runs using all positions of an instrument. So you collect the results as percent prednisone dissolved at 30 minutes from 2 consecutive runs on the dissolution assembly. You calculate the overall geometric mean and the mean of the variances of both runs, and then you compare the obtained results with the applicable acceptance limits. So if the PVT passes if both values meet the acceptance criteria, so it's the overall geometric mean and the mean of the variance of both runs that need to pass. So from experience, the most common causes of failure are procedural. So you have improper degassing, you have inaccurate dispensing of your prednisone, resident sampling probes and or thermometers in the vessels. Then the sampling technique, so you're doing manual sampling there. So the other is more equipment, and this is actually the purpose of the test, right? You're not wanting to test the technician that's doing the PVT. You're trying to test your equipment. So reasons there could be poor quality vessels, the apparatus alignment, and vibrations, right? So what has changed over the years, right? So prior to 2006, the USP performance verification test or PVT using the prednisone and salicylic acid was the only option available. But since then, since June, the FDA published the MQ procedure. You have an FIP position paper on mechanical qualification. What else happened? The salicylic acid was continued for like the PVT. The FDA MQ guidance for the industry was published. So there's a clear trend there to mechanical qualification, you have the USP Dissolution Toolkit. So a lot of things have happened. So the ICH guideline. What else? The ASTM, MQ procedure, and the ASTM MQ procedure included in the Chinese pharmacopoeia. So you can see over the last 15 years, or in this case 10 years, really a clear trend to make mechanical qualification more established for your dissolution equipment. So here's a key statement from the regulatory guidance on the MQ guidance for industry. Either the USP procedure or an appropriate MC method executed according to a written procedure will satisfy the GMP requirement for calibration of the laboratory apparatus. So now, I think that's great. You have the freedom of choice what you want to do. Right, so there are two distinctly different approaches to dissolution qualification. The USP prednisone test is a holistic system check, if you like, and the FDA ASTM approach is a more process-oriented and does not include a chemical test. Right, so for your MQ, you're not using any tablets. Instead, you're having tighter tolerances and you need additional SOPs. Work to monitor your dissolution environment, right? So today's qualification environment, as I said, you have the PVT. It has great advantages. So it's a holistic test with controlled tablets, and it can detect issues with your vessel quality and environmental issues like vibration, for instance. The MQ advantages, so your mechanical qualification, really is the time to perform. You can do that at a greater frequency. You are already doing most of it, like you would always do a check of your dissolution tester before you start your tests, and it is now widely acceptable, right? So, I mean, with the time to perform, I would say I can do an MQ on a dissolution tester in like 30 minutes. For the PVT, you would probably need something like half a day or a full day. Greater frequency, I like that fact because theoretically, if you have the right equipment, you could do an MQ of your tester before every dissolution run. Now that is maybe going a bit over the top, but you could do it, let's say, every week, every month, just to check that your dissolution tester is acceptable, right, and giving you the right performance. So what are the disadvantages? Time, obviously, for the PVT. It doesn't specify the problem, right? So even if you have a failed result with your PVT, you then have to start searching where this is coming from. The expense, so you have to pay the person that is doing the PVT, and it takes long, so you have the instrument downtime. And you also have the cost of materials because you obviously have to buy the calibrator tablets, you have to degas your water, or these kind of things. Now with the MQ disadvantages, you do have a transition process that you also need to document, right? Why are you transitioning? It does not show you if you have problems with your vessel quality, with your vibration. With your vibration, I must say, it's only because you don't have a spec for it. If you do have the right equipment, you can actually show that there is vibration. This can be measured, right? And laboratory environment issues, this is obviously also not shown. So if you have a person, doing handling errors and things like that, that is also not being shown up. So what are the wrong reasons really to consider a move to mechanical qualification is the apparatus will not pass the PVT, right? You cannot just do a PVT, see it fails, and then you do an MQ and say, oh, now it passed. You must diagnose and resolve the issue that you've seen during the PFT, and you need to close out your PFT, like you have to pass prior to moving on, right? What are also wrong reasons? You're not willing to replace your dissolution accessories. So you see a really bad example there of a dissolution pedal where the coating is flaking off. So you have to take good care of your pedals, baskets, and vessel. And worn components can lead to PVT failures, right? So what are the right reasons to consider a move to MQ? You have a method for dissolution process improvement and better monitoring. As I said, you can just do that as many times as you like because it's so fast. You can connect your instrument qualification to daily dissolution activities and improve components and system quality. So you can also have like a monitoring system and see like, you can do some kind of trending and see if there's anything going wrong with your dissolution equipment over time, right? You ease the burden of potential failure investigations because you have a better visibility to instrument performance. And the sensitivity of prednisone is no longer a subject of discussion, right? I mean, there's been big debates about the quality of these tablets. That sort of people didn't like the fact that, sort of new stability studies were done and then sort of the specs were changed, all these kind of things, right? So what is available on the market? So you have individual tools or gauges. So you have substitute shafts or adapters that can be used. It's quite time consuming still. You have multiple tools to calibrate. And there's a visual interpretation required, and that leads to variability, and you have to do some form of manual documentation of all this. So what we offer here from Agilent is it's called the 280DS mechanical qualification system. So you have a vessel module that will then sit in your vessel, and with sort of lights inside the instrument, you can measure a lot of your parameters. You have an instrument model that would be sitting on your vessel plate, so you could measure the level. And what I think is great, you can also measure vibration. You have a temperature probe, obviously, to measure the temperature of your water bath or the media in your vessel. Now, that whole instrument setup is connected to a PC, and it comes with software. So the one great thing is the software will guide you through your mechanical qualification. It basically tells you what you have to do. It's very easy to use. But the other benefit is you will get a report that you can file wherever you like in your database, in your LIMS system. And that database is an SQL database, so you can't delete any of your data. So you can also use this in your 21 CFR compliant environment. With the report, as I said, you have full traceability, you have the electronic signature function, all the benefits you can think of. So yeah, I mentioned that. So you have the vessel module with the sensors. You would measure the rotational speed, the basket and shaft wobble, vessel centering, shaft verticality, vessel verticality, basket pedal height, with that little arm in the bottom, vessel plate level, and the temperature. That would be with the other modules. Sorry. So it's time saving, as I mentioned, to do a mechanical qualification of your tester. It takes about 30 minutes, really. You can do shorter calibration intervals to narrow the scope of your potential failure in your investigation. So imagine instead of, looking at all your data from the past 6 months, it would now only be just a week, maybe 2 weeks or a month. You can sort of do a trending and identify instrument problems and monitor these variations over time. And there's no guesswork or manual documenting values because the values are all documented by the software, and you get a report in the end, right? And you don't have to purchase any standards or tablets. It can be a daily tool for failure investigations. So what are the conclusions on that topic? So there are two equally valid dissolution qualification techniques that now exist. And I must say a lot of our customers, they are still doing both. They would do a PVT every 6 months, or maybe, and then in between they would do mechanical qualifications more frequently. It's up to you which route you decide to take, and it's up to you how often you want to qualify the equipment. Just check out which route really works best for your labs and for your systems and products, and don't just swap over techniques with no explanation. Document your decision and what steps you've taken to cover aspects of the solution qualification that are covered by a PVT, which are not covered by MQ. And these new tools, they provide you very fast and efficient methods to perform your MQ. So further on from this, we're coming to another interesting topic, and that is data integrity. So creating a paperless compliant dissolution testing environment. So really the question here, is data integrity a new regulatory expectation? Of course not. It isn't, right? But the FDA certainly has changed the focus of data review processes. You can see that it is no more just a review of paper printouts of results and reports. It's no more a review of just result sets. It's no more a review of just the reports, right? It's a lot more. If you have an audit and a review is happening, then what the reviewers look like really is the source data in the electronic system, the associated metadata, audit trails is a big focus of the FDA, server activity logs, operating specific activity logs, result sets, application specific activity logs, instrument error logs, IT tickets to check backend database changes. So a really sort of holistic review that is now happening. So just a sort of explanation how you come to get a warning letter, and I hope no one of you gets a warning letter. So during an audit, there are observations and concerns, and you try to resolve them there on the spot during the audit. If it cannot be resolved during the audit, you will get a Form 483 at the end of the audit, and you will have a company response within 15 days after the audit. So what happens then with this response? The FDA will review this. They will look at your track record. They will gather all other information. They will look at the Form 483 and the company response. If there are still serious deficiency, the company will get a warning letter. So focus on data integrity issues. If we look at the years from 2005 to 2010, then it was sort of the calibration of the instruments. Data integrity was up there as well, but calibration was even higher. And the HPLC usage, there was repair, medical devices, some method validations. Now this changed from 2011 to 2015. A clear focus here on data integrity, right? So what are common data integrity issues? And I mentioned a couple here. We also have them in a white paper that is available out there. And really, I mean, we put that, or Paul put that in the white paper there, and he didn't make this up. This is really sort of the real world. So shared passwords is a big issue. The analysts share the passwords, and it's just not possible to identify or who created changes of the records. User privileges, the system configuration for the software does not adequately define or segregate user levels, and users have access to inappropriate software privileges such as modification of methods and integration. Computer system control, laboratories have failed to implement adequate controls over data, and unauthorized access to modify, delete, or not save electronic files. It's not prevented, right? So the file therefore may not be original, accurate, or complete, right? Processing methods, this is more for the HPLC, so integration parameters are not controlled. Incomplete data, the record is not complete in this case, and the definition of complete data is open to interpretation. Audit trail is always a big topic. In this case, our laboratory actually had turned off the audit trail functionality within the system, and it's not clear who has modified a file or why. So when we get these data integrity surveys from the customers, this is obviously always one question is in there: can you turn off the audit trail, yes or no, right? So what are the GMP requirements in that light? So all laboratory equipment must be validated, all methods must be validated, and you must have a computer system validation. Your data integrity should be according to the 21 CFR Part 11 or Annex 11 guidelines. You want to ensure an appropriate level of employee qualification and training. You want to define specifications for lab equipment and reagents, define SOPs for sampling, sample labeling, etc. You want to have SOP documents to manage out of specification situations, and you want to do regular self-inspections and internal audits from an independent quality assurance department. So what's the FDA stand on the 21 CFR Part 11? It says, like, these regulations which apply to all FDA program areas are intended to permit the widest possible use of electronic technology compatible with FDA's responsibility to protect the public health, right? So that is the FDA's main goal, protect the public health, and they see that data integrity is a very important aspect of this. So some background, I don't want to go into all of this. You will get that for your records. 1997, the FDA publishes the title 21 CFR Part 11, Electronic Records and Electronic Signatures. The companies were really sort of unprepared for that procedurally and technologically. That's why in 2003, the FBI published this guidance for Industry Part 11 and explain sort of clarification on scope and enforcement of this Part 11. So sort of helping there for a greater understanding. Then there was the guidance for industry on computerized systems used in clinical investigations, and also you have the Annex 11 to the EU GMP guidelines for computerized systems. So in summary, what does the 21 CFR Part 11 actually cover? It covers 3 specific elements of your laboratory's operation. So the security of electronic records, the attribution of work, and electronic signatures if they are used. So now the big question really is, how can we apply this to our dissolution environment? And what can we use to make sure we are prepared for an audit situation, right? So here's an example that is, again, a real-life example. We have this dissolution hotline. So this is a question that we got from one of our colleagues. So the question was, is a standalone 708DS, so that is our apparatus 1 or 2, with manual sampling, operated via the built-in controller, that is a touch screen, fully FDA compliant? So 21 CFR Part 11. Is there any advantage in operating from a dissolution workstation? That's the software. Without even an auto sampler, and does that make regulatory compliance easier? So original question there. And our reply to that was, so by the definition, no firmware or software like the dissolution workstation that we're going to see in a minute is compliant on its own, right? Rather, these should be considered tools to facilitate compliance. So now already the 708, just a dissolution tester, has some features that can help you to comply with the rules. So there's a built-in printer for documentation of actions. You have different user levels for controlled access and privileges, and you have individual usernames and passwords, right? So if you now add the software, that makes compliance a little bit easier, because what you have now, you have again user levels for controlled access and privileges. You have individual usernames and passwords. You have every system action and or change is documented in the audit trails. You have electronic signature capabilities, and the data is stored in a secure database managed by MS SQL. So the answer is yes, a laboratory seeking a compliant and paperless solution would benefit by using the dissolution workstation software. So I'm going to give you a quick run through the software that we have. So we call it Dissolution Workstation software. It's quite flexible. You can connect up to 4 systems. You can have them online, up to 4 systems online at any time. It's compliant in As I just explained, it's not compliant by itself, but it can be used in a compliant environment. It's efficient. You have a paperless solution there, and it's secure. You have integrated data security with easy export features. So let's take a look at what this is. This is your sort of your user interface with a method management that you can see up here. So you can create your method and run method, and you get your test reports. What else? You have a system management. You will create configurations for your dissolution equipment, as many as you like, really, but you have separate files, and they're created and maintained for your dissolution systems, and as I said, for your methods. Then you have a central database where all this data is stored. Your test, once it's running, is displayed in real time, and all the system activity are recorded. So you will have a run log in the end that tells you exactly what happened on the system. You have complete audit trails, e-signatures, and user level to control the access. So, and then what you see here with the different versions, so whenever you create a method or a configuration, you make any changes, it's also controlled via your versions, right? And you have different user permissions to help with that. So just looking at the audit trail capabilities here, I mean, it can do a lot more, but just to give you an example, so you could look at that. You can see that I logged onto the system, I terminated the application, then I logged on again. So the time is there, date and time stamp, the activity, the workstation I did this with, and my user ID is there as well. Data verification, that's possible as well. So you just want to, once you've just created a data file, you want to make sure while this was created, no other system like on the PC was sort of getting into your data file and was able to do any manipulations. This is while you're running your dissolution test, you kind of get a real-time status of all your active dissolution systems. You have here on the right your sample collection status, if you do have a sample collector. And now look at that. You can even monitor your vibration, right? If you have a 280 instrument module connected. So that can be done. You're tracing your RPM, your bath temperature, the vessel temperature, and you get a real-time activity log as I'm monitoring is there, and then in the end you will of course get a report for your system, right, with your time points, your bath temperature, your speed, your vessel temperature, the elapsed timing of your test. What is done in terms of user security? Of course, you have login access. I mean, otherwise this wouldn't work. You have then user levels, right? That's what you can see here on the right. So you can give a certain user different access options. So you can, for instance, have somebody like a VK Disso run method. So you can have a user that could only run methods, right? This person cannot change any methods, for instance, right? You have another person that might be the laboratory manager that can do method maintenance, right? So that person would look at methods, would set them up, or these things. Then you would obviously have a database admin as well. You can lock the application, so it locks itself after a certain time, but you can also lock it and go away. So just to make sure nobody else can access your equipment. So in summary, the Dissolution Workstation software, it really simplifies your method and system management for dissolution testing. You centralize your data capture. Everything is in your centralized database. It actually works also in a server client scenario. You record your essential test parameters at user-defined intervals. It facilitates your 21 CFR compliance by implementing security, record retention, and audit features, and it just helps you to be ready when an audit situation arises. So a little bit sort of on further information, if you want, from Agilent, you can go and visit the Dissolution community for links to recorded webinars, frequently asked questions, white paper, all these kind of things. You can contact us on the Dissolution hotlines. It's about 5 or 6 people answering questions on the Dissolution hotline, and that can be anything from like asking for a part number to like saying, look, I've got a new product, what's the best way to go about method development for this product. So that was it, my presentation on the Dissolution Toolkit. So thanks a lot for your attention. I'm going to stop sharing my screen and look what we have in terms of questions, if there are any.
Alrighty, wonderful. Thank you so much, Karen, for that fantastic presentation. All right, everybody, so we will go ahead and go to our Q&A portion concerning today's presentation. If you have any questions, please feel free to place them in the Q&A box located on your WebEx platform. If you would like to ask a question over the phone line, you can click the raise hand icon, and then I'll be able to unmute your line. As a reminder, I will be sending the recording of today's presentation as well as information to register for next events in the series, usually within 24 to 48 hours, so everyone should be receiving that sometime tomorrow or the next day. All right. Our first question here reads, does the 280DS need to be calibrated, and can I do this myself?
Well, thank you. And yes, the 280DS, because it's a tool for mechanical qualification, it does need to be calibrated. Unfortunately, you cannot do that yourself. So, but we do have a calibration service, and we have 3 calibration centers located throughout the world. The reason basically why we say you can't do it yourself is because we have about 50 tools and fixtures that are needed for the calibration of that instrument, and you cannot simply set this up yourself. Yeah.
All right, wonderful. Thank you. We've got one more question here. Do you see a trend in dissolution testing to more automated solutions?
Yeah, there is a clear trend, especially now with the current corona or COVID-19 situation. We see that quite clearly that customers sort of, recognize like, oh, what do I do with my dissolution testing if I can't have all my staff in the lab all the time? So we clearly see a trend to dissolution testing and the flexibility to start your tests in the evening, have them finished in the mornings, clean up the system, start the next run, have them run over the weekend. So clear trend there to automation, yes.
Perfect, thank you. All right, I don't see any other questions in our chat or Q&A box. So if anyone else has any other questions, please feel free to place those in there now at this point. But for now, it appears we have reached the conclusion of our event. So please join me in thanking Karen Kroll-Golmer for her wonderful presentation.
Karen.
Karen, yeah. You guys know. To go over a couple of points, so please note this is an ongoing series. You can see our next 4 events listed in the slide currently displayed. And then this is going to be the URL where we house all of these events. You can check out previous recordings for on-demand and then register for upcoming ones. All right. Alrighty. Well, I don't see any other questions in the chat or Q&A box, so we will go ahead and start closing out this session. I will leave this open for a few minutes to receive any feedback or additional questions, but we are now going to close the audio line. Thank you all once again for attending, and on behalf of Agilent Technologies, we wish you all a great day.